Chapter 5, Prokaryotic Genetics. Genetics is the study of heredity. It's the study of what genes are, how they carry information, how information is expressed, and how genes are replicated. Genes is a segment of DNA that encodes a functional product, usually a protein. A genome is all of the genetic material in a cell. Genomics is the molecular study of genomes. A genome is organized into chromosomes. A chromosome is a structure containing DNA that physically carries hereditary information. The chromosomes contain the genes. The genotype is the genes of an organism. For example, the sequence for the gene that codes for the flagellin protein in bacteria. The phenotype is the expression of the genes, and that's when you actually express the flagellin protein that you find in the flagella. Flow of genetic information. So here is your parent cell. What we're going to do today in this first part of Part A of Chapter 5 is we are going to focus on the DNA replication. Like you learned back in Chapter 2, we have to replicate the DNA before we can go through binary fission, in which we produce two identical daughter cells. So we're going to take a look at the replication. And Part B will then be focusing on the expression portion, in which we're going to go through transcription and translation to make a protein. And then finally, in Part C of this chapter, or part, we're going to be looking at recombination, how information is genetically exchanged between two different cells. So let's go ahead and begin with the replication portion. DNA is a double helix associated with proteins. It's a polymer of nucleotides. Each nucleotide has a nitrogenous base: adenine, thymine, cytosine, or guanine. This is all found in the deoxyribose phosphate backbone. So here you'll see the sugar, deoxyribose. Here you'll find the phosphate groups. That's your backbone. The strands are held together by hydrogen bonds between the A and the T and the C and the G. So we know now that the A goes to T and C goes to G. So these guys are going to have hydrogen bonds. So that's those dotted black dots that you see there are your hydrogen bonds. The strands are what we call anti-parallel. That means one strand runs 5' to 3', and the other strand runs 3' to 5'. So if they were parallel, then they'll both be running 5' to 3'. But this one is going 5' to 3', and this one is going 5' to 3'. Complementary, meaning that the base sequence of one strand determines the sequence of the other strand. This allows for precise duplication. So as long as I know what the first strand is, as long as I know that there's a T, a G, a C, and an A, I can match it up. So T goes to A, G goes to C, C goes to G, and then A goes to T. There's also diversity in DNA. There's only 4 kinds of bases, but each gene contains about 1,000 of these bases. So they can be arranged in 4 to the 1,000 different ways. The bacterial DNA is usually a single circular molecule that takes up 10% of the volume. We know that we can have about 4 million base pairs, so 4 times 10 to the 6 base pairs, and it's about 1 millimeter long. In DNA replication, one double-stranded DNA from the parent leads to 2 double-stranded daughter molecules. One strand is going to act as a template for another, complementation. DNA replication goes through something called semi-conservative replication. You're going to have one old and one new. So we're going to separate these parent strands, and what we're going to end up having is going to have one parent and one daughter, one parent and one daughter. So the two parents will never go back together. The DNA is copied by an enzyme called DNA polymerase. DNA polymerase is the enzyme that adds new nucleotides to the growing DNA chain. It can only add to the 3' end. So there are additions to the 3' end of the DNA strand in the 5' to 3' direction. So what are the steps of DNA replication? Well, 1, the parental strand is unwinded by enzymes. The replication fork forms at the origin of replication. The leading strand is synthesized continuously in a 5' to 3' direction towards the replication fork. The lagging strand is synthesized in pieces, also in the 5' to 3' direction, but away from the fork. All of this in the lagging strand is initiated by an RNA primer. An RNA primer is needed to start because DNA polymerase can add to the 3' end. The pieces that are formed are known as Okazaki fragments. The RNA primers are removed by DNA polymerase, and the Okazaki fragments are joined by DNA ligase. So let's put this together in one picture. So here is my parental strand. The first step is we're going to unwind that parental strand. And as we unwind it, we're going to form our replication fork at the origin of replication. There's one strand up here, and there's another strand here. Remember, DNA polymerase can only copy in a 5' to 3' direction. It has to add to the 3' end. So if you notice, this strand is going 5' to 3'. This one is going 3' to 5'. So if I am DNA polymerase, I am going to start from this end of it. I'm going to start adding at the 3' end so I can go 5' to 3'. And this is going to be your leading strand. And the leading strand, again, is synthesized continuously in the 5' to 3' direction towards the replication fork. Let's take a look at the other strand. The other strand would then be my lagging strand, and it's running 3' to 5'. So that means the DNA polymerase is going to be going in this direction to copy in a 5' to 3' direction. But the lagging strand is synthesized discontinuously in pieces. So you're going to have little pieces being made, and each one of those pieces is called the Okazaki fragments. The lagging strand is initiated by the RNA primer. So here's your RNA primer, and then it's going to allow DNA polymerase to start adding nucleotides. And as DNA polymerase is adding new nucleotides, it's removing the RNA primer. Once we've completed all of the pieces, then we're going to use DNA ligase to seal it. Okay? DNA replication is bidirectional. That means that in humans we only go in one direction. But with bacteria, it can go in two directions. So here's one replication fork, here's another replication fork. So you have two replication forks, and they all move in a closed loop. So you'll see that it's moving around this way, and it's moving around this way, till eventually we all come back to the same point again. And this is why bacteria grows a lot faster. Proofreading. Replication is extremely accurate because of proofreading by DNA polymerase. Here ends part A of chapter 5.